Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast, formerly the Future Tech Podcast. My goal here is to find the top people in their professions. This could be 1 in 100, 1 in 1,000 people that go above and beyond. They don't just do enough to be licensed. They want to be really good at what they do, and they are. So my guest today is Jennifer Bomberger. She's an associate professor at University of Pittsburgh in the Department of Microbiology and Molecular Genetics. And we're going to be talking about microbe and virus interactions, looks like specifically in the respiratory tract. So this should be very interesting. Thanks for coming, Jennifer. How are you doing?
I'm doing great. Thank you so much for having me. I'm excited to chat today.
Oh, good. So tell me a bit about your research in your own words.
Yeah. So I guess my research is founded under the principle of trying to understand why patients get chronic bacterial infections. And this is often the patients we're thinking about are patients with chronic lung diseases. And so in my lab, we focus a whole lot on cystic fibrosis patients and the chronic infections that they develop. And so they're very well known to develop a number of different respiratory infections throughout their lives, but by far the most common is infections with bacteria named or called Pseudomonas aeruginosa, as well as Staphylococcus aureus. And so we focus a lot primarily on Pseudomonas aeruginosa in my lab and why these infections are caused, how they're caused, with the goal of trying to prevent them in the future. And so it seems like when we look at clinical studies that patients with Pseudomonas aeruginosa infections, that's most correlated with worse lung disease in patients and disease progression. And so that's why we're trying to understand why these infections are caused and the goal of trying to stop them. And so
a quick question, where in the lungs do these bacteria hang out? Are they in the trachea, or are they down in the alveoli or the bronchioles?
Yep. So you can find these bacteria in the sinuses, which I think we're going to probably touch on a little bit because we've become really interested in sinus disease recently. So they're in the sinuses as well as down in the large airways. Is where most of the infections in cystic fibrosis patients are in the larger airways. And so only in very late disease courses do they end up with infections in their alveoli. It's more in the larger airways. So like the bronchioles, a little bit in the trachea as well.
And what is the natural healthy microbiome look like versus the microbiome of someone with cystic fibrosis or one of these chronic infections?
Yeah, so that's a super controversial area. So this is something that is debated heavily in the field if we have existing microbiomes in our respiratory tract. And so I can give my opinion of where I come down on this, but you can find people on different sides of this. And so I think if you look at So one thing you have to know kind of before having this conversation is that in cystic fibrosis, one of the major defects is a defect in this chloride channel called cystic fibrosis transmembrane conductance regulator, or actually it's an anion channel. So it conducts both chloride and bicarbonate. And what it does is maintains the proper hydration of your airways, which then allows your airways to have effective mucociliary clearance. And so you need to have hydration so that the cilia that are on your airway epithelial cells can beat and can clear debris and pathogens out of your respiratory tract. And so for healthy individuals like you and I, we have effective mucociliary clearance. If we inhale either pathogens or pollutants or things into our lungs, our respiratory epithelium will use the mucociliary escalator, it's called, to kind of clear the debris up the respiratory tract, and then we swallow it, and everything, if there's any microbes in there, they get killed in the stomach by the acidity. And so in healthy individuals, where I come down in the microbiome debate is that we're constantly microaspirating, and we can aspirate bacteria from our mouths, from our upper respiratory tract, and from the environment into our airways. And that if you sampled us at any second, there'll be a few bacteria in there that are just in the process of being cleared out of our respiratory tract, but we have very few bacteria if we were to sample you or I. Whereas cystic fibrosis patients, they can.
So you don't think that there's a natural microbiome or microbial attachment in the lungs? Like, are there bacteria there that are getting something in terms of food? Is there a niche that they're preferentially occupying on a regular basis, or it's just temporary?
I do not believe there is. I believe they're transient. They're transient constituents that are there and they're on their way out. In the nasal, like in your nasal passages, you are colonized with bacteria there, but those are quite different than what we see in the respiratory tract. And so a lot of the bacteria that we see that we think of as, I guess you could call commensals in the respiratory tract to the microbiome, would be Neisseria species. They would be Corynebacteria, Moraxella, things that actually they tend to be bacteria that we find in the mouth, different streptococci. Bacteria that we find in the mouth can be, and we think probably are being aspirated into the respiratory tract, and that's in the airways, and that's how you see them if you would sample a healthy individual for a microbiome. But the bacterial burden is much, much lower than a patient that's actually colonized with the bacteria. Does that make sense?
Yeah, and that's why I interrupted you, but you were probably about to say in CF patients, I guess they can't clear mucus effectively, so the bacteria then take up residence. Correct.
Yep. So CF patients, because of this defect in this anion channel, they don't have effective mucociliary clearance, and so when they inhale or have bacteria in the respiratory tract, they can't clear it. And then because of some of these other defects then in the mucus, the actual like how the mucus is made up in cystic fibrosis is different than ours. And so for a whole lot of reasons, they can't clear the bacteria out. And then those patients will have, they have bacterial burdens up to 10 to the 10th, 10 to the 9th bacteria. So enormous bacterial burdens in their respiratory tracts for decades. And so they have real serious, like they have a very clear microbiome where ours I think of as a transient, but that is something that's definitely debated in the field, and different people have different opinions.
Well, what's the consequence then to people with these chronic, lung infections? Is there anything beneficial about it? And if it's negative, what is? What are all the negative consequences? Like, how do they affect the functioning of the lungs and the exchange of air, and et cetera?
Yep. So I don't know that we know of any good things that come of it. I think it's all negative. And so I think when we see lung function decline in patients, I'm talking now mostly about cystic fibrosis because we know a lot about those patients. If you look at the lung function decline in those patients over time, we think it's probably partially contributed by the bacteria themselves and things they're doing, toxins and different substances they're secreting that impact the health of the respiratory epithelium in your respiratory tract. And then the other side of that double-edged sword is that those bacteria that are then resident are also recruiting in immune cells. And cystic fibrosis patients are known to have really strong inflammatory responses in their respiratory tracts, but they're completely ineffective. And so they recruit in neutrophils and macrophages and these immune cells that that seem to be an appropriate immune response. They should be recruited where you have a bacterial infection happening, but then the immune cells in cystic fibrosis are not effective in actually clearing the infection. And so then they do what they do. They produce toxic substances, which they should be doing to kill bacteria, but it doesn't work properly in cystic fibrosis. And so those toxic substances end up just causing damage to the respiratory tract. And eventually you have scarring and tissue damage in the lung. And that's caused probably as much by the immune response as by the bacteria, but the bacteria are the ones that have recruited that immune response in. And so those infections are really problematic for patients. And so, and then they have these infections for decades, and so the bacteria, we know, evolve and really become fine-tuned to living and persisting in that respiratory tract environment. And so they become very, very hard to clear with our current therapeutics.
Well, what about fungi, protists, and then especially, bacteriophages? Are you looking at the presence of those and their interactions?
So we are not looking at a number of those other microbial constituents at this point, but my laboratory looks at, we're starting to look a little bit at bacteriophage, but more therapeutic phage and how they change the microbial communities in the respiratory tract. But our main interest is in how respiratory viral infections, so an acute viral infection, how that might change the respiratory microbiome. And so that's been the major goal of my lab since I started my lab. So fungal infections, I think, are going to turn out to be really important in CF, but that's not something we're studying. There are great labs doing that work, just not us.
So how does a viral infection of the lung respiratory tract look different from a bacterial one?
That's an interesting question. So I guess when you get a respiratory, when any individual, healthy or with chronic lung disease, gets a respiratory viral infection, it triggers a different type of immune response. And so we have these things called pattern recognition receptors that are in our immune cells and in our respiratory epithelial cells that their whole role is to detect patterns from microbes. And so they can detect what sort of microbe is infecting, and then they trigger different types of immune responses that are supposed to be tailored to killing that particular type of infection that they've detected. So if they detect an extracellular bacterial infection, they respond in one way that would be able to clear an extracellular bacterial infection, presumably by calling in the right types of immune cells to clear that infection. If they detect an intracellular infection, sometimes those things are triggered by trigger different types of responses where you would want to either kill that particular epithelial cell so that that microbe can't replicate, or there's very different responses that happen. And so because respiratory viruses need the respiratory epithelium to propagate, it's kind of when the host detects the respiratory viral infection, they initiate a specific immune response that normally shuts down gene expression or shuts down protein translation in the epithelium, partially so the virus can't make more virus particles, but it also causes systems to be put in place where you start sequestering nutrients because microbes need nutrients to replicate, so the host should be sequestering nutrients to prevent further infections. And so there's a cascade of events that happen when you detect a respiratory viral infection that allows the host to try to contain that infection.
What do you mean? What's sequestering nutrients? The epithelial cells, and for what?
The epithelial cells. The epithelial cells are sequestering nutrients, and so nutrients like amino acids or like metals, like iron. Iron is a cofactor. Iron is of particular interest to our lab, which I can tell you more about. But iron is an important cofactor for enzymes in our body that they need to function. And so the host and bacterial enzymes also need iron. So iron is a major source of competition between bacteria and the host. And so that's one thing that can be competed for during a microbial infection.
Well, microbial infection or viral infection?
Sorry, so that the iron example that I was giving is for a bacterial infection.
Okay, so if there's a bacterial infection, at least so far to me, it makes sense that the cells would sequester certain nutrients because then they get them and the bacteria don't, to starve them out. Mm-hmm. With viruses, what would be the point of sequestering nutrients? Is it because
that ends up being more of an energetic? Sorry, that becomes more of an energetic. So normally metabolism changes in a way. So that's slightly different. Maybe I was broadly explaining nutritional immunity for lots of different types of microbes. For viruses, it normally tends to be changes in metabolism in the host cell that kind of, and again, like I said, they end stopping translation to try to stop like processes in the host cell that allow the virus to replicate.
Oh, okay. But so it's primarily a defense against replication, but are there any other kinds of defenses when there's a viral infection? So
sometimes there's changes in the host cell. So it depends on the virus. So some of the, so one of the things I guess I should explain at the beginning, when the host epithelium detects a respiratory viral infection, it initiates a signaling cascade. It's called a cytokine called interferon. And that interferon elicits a signaling cascade in the host cell, which we call the antiviral program. And so it turns on hundreds of genes in the host cell that then allow the host cell to kind of start preparing to defend against a viral infection. So depending on what the virus's replication mechanism is, the host often, one of the mechanisms they could do, I can give an example, would be you would change lipid metabolism. So for instance, if the virus needed host lipids to be able to form an envelope or something to be able to actually be secreted, the host can then manipulate lipid biosynthesis so that those lipids are no longer available for the virus. And often there is a competition. The virus often is co-opting those mechanisms to try to make more lipids, and it's kind of a battle between the host and the virus to try to make what the virus trying to make what it needs and the host trying to not make those things to try to prevent against the infection.
Well, if for instance, the epithelial cells are doing this, where's the communication happening to signal them to know what to do? Is it happening where, are the immune cells, I guess, eating some of the viruses and then they're communicating through extracellular vesicles to the epithelial cells, here's what to do? Or, how do you think this knowledge transfer is happening?
Yeah, so I don't know that we know entirely how this knowledge transfer is happening. There's definitely, so we think of the epithelium as normally being one of the first, in the respiratory tract. The epithelium is often the site of a lot of respiratory viruses, their site of replication. And so the respiratory epithelium is typically where these viruses will be initially detected. And then the epithelium then will send out signals to other cells. And so one of those signals is interferon. So it makes this cytokine interferon that can communicate with immune cells, and it can communicate to other epithelial cells to kind of put the whole epithelium on lockdown, sort of, to know that there's an infection around. So cytokines are one important way it does that. There is this idea of immunoeducation or communication through extracellular vesicles. And so this is one potential mechanism by which the host reaches out to tell other cells what's going on in the host cell. There's been interesting studies on that, not work that my lab has done particularly in that area. We do have an interest in extracellular vesicles and how the respiratory epithelium can communicate to bacteria, or how bacteria actually probably are co-opting this host communication mechanism. And that interaction, I think, is interesting. But for the immune cell communication, that's something that's still being worked on in the respiratory tract, but is likely a mechanism for communication based on what we know from other mucosal sites.
So what happens now if a CF patient, they have a persistent bacterial infection and the bacteria just, they attract bacteriophages that are trying to prey on them. Now you have, a persistent viral component, supposedly only attacking the bacteria, and you have a persistent bacterial component. Can you look at the immune response and see if there's a signal there that says, the host recognizes that there is a persistent viral threat or that there are viruses there, even though they may not be attacking the host cells?
So I think, are you distinguishing a mammalian respiratory virus from a bacteria, like a bacterial virus, like a bacteriophage, or are you using that virus term interchangeably, just so I understand what the question is?
No, it's fine. I'll be more specific. So if I have CF and I have a persistent bacterial infection, those bacteria are going to attract bacteriophages, viruses that will prey on them. I'm sure naturally there'll be at least some. So even if those viruses don't affect the host, they're certainly going to kill off a lot of the bacteria that are inside the person with CF. But does the person themselves recognize that, there's viruses here, even though those viruses aren't attacking, my cells, they're still attacking bacteria, but they're still there, they're still present. And then does the host prepare any defenses against them, or are they invisible to the host because they can recognize somehow, these viruses are not going to hurt us, we don't care?
That's an interesting question. Yeah, so that is a very current and new area of interest in my laboratory, is trying to understand, does the host actually recognize bacteriophages that are in the respiratory tract? And so we are still in early days of our studies, but I can tell you what other folks have done. And so there's a really nice paper out of Paul Boyke's lab at Stanford. And so they're looking at, their particular interest is in filamentous phage. And so these are phage that are actually promoting Pseudomonas biofilms, but they are bacteriophage that are in the respiratory tract. And they are able to be detected, they believe, from immune cells in the respiratory tract, and initiate immune signaling. So they can be detected, and they can trigger immune responses in the host. So that's something I think we are super interested in, but we are in very early days of trying to start those studies to try to understand. So it's been shown for filamentous phage that are made by bacteria. We're interested in lytic phage. So these phage that we're treating to patients are those phage that we're putting in the respiratory tract of patients to kill their bacteria. Are they detected by the host, and do they trigger some kind of immune response that could be either protective or not protective? We don't know. And so those are studies that we're just starting right now in our laboratory. So it's a great question, and too early to talk about publicly or to say the answer yet. So I think
that's fine. Well, if someone has CF and you do find a phage that kills the bacteria, the Pseudomonas originalis, whatever kind of Pseudomonas they are, then I would guess you still have the problem of getting them out of the person's respiratory tract, but maybe there's a mechanical vibrators that, have the person cough up a whole bunch of mucus and that work? Like, is there a problem even if you're able to kill those bacteria?
Yeah, so I think that's a great question, and we don't know. So I think there's some things we still don't know. So we don't know. So there's been some studies out of several different laboratories. We're collaborating for our work with Paul Turner and John Koff at Yale, who's been doing some of this work with giving Pseudomonas bacteriophage to patients in their CF clinic, and they published some of this work. But they've seen efficacy in treating patients, which is really exciting. We've got these patients that have incredibly multi-drug resistant infections with Pseudomonas aeruginosa, and there are no antibiotics left to treat them. And so it's really important to come up with these new therapies. And so they've seen really exciting results in reducing bacterial burden in these patients. But from the microbiology side, we don't know if these phages are actually able to penetrate into the bacteria. So one thing I guess we should introduce in this conversation is the concept of bacterial biofilms. And so bacteria growing in these bacterial or microbial communities in the respiratory tract, we think bacteria, when patients, like I said, have these infections for decades with Pseudomonas aeruginosa, we think one of the reasons they become so antibiotic resistant is they grow as these bacterial biofilms. And so these are communities or aggregates of bacteria that when they grow together, they change their gene expression, they make matrix, they become incredibly antibiotic resistant, and our antimicrobials no longer work well on them. And so by making this matrix, it kind of gives them the structure of this little ball of bacteria, sort of is what they look like in the respiratory tract. By making this matrix, they make it very hard for a lot of antibiotics and things to penetrate. They can put charge on this matrix so that things can't penetrate well into it. And so one thing we don't know, and we've been talking, is something we would like to study is we have models in my laboratory where we can grow these bacterial biofilms on respiratory epithelial cells and use imaging to kind of watch what the dynamics of the biogenesis of those biofilms, and then apply treatments and try to look at how it changes the biofilms. We would like to know, when you treat with phage therapy, is it actually penetrating into those biofilms? So often patients aren't completely clearing their infection, and so we don't know if the phage are not penetrating, or like you said, do they penetrate and we just can't clear everything out of the respiratory tract? There are some of these things that we still don't know.
Okay. Yeah, that's true. I forgot that the bacteria form biofilms.
Mm-hmm.
So
that's actually a major interest.
Yeah. Has anyone been able to, in the lab in a dish, set up epithelial cells and have these bacteria grow in them and observe what the biofilm looks like, or do a biopsy and somehow preserve the structure of the biofilms that come out and see what that tells you?
Yes. Yep. That's a great. So folks have, for quite a few years, we've gotten clinical samples from patients and been able to look and see these aggregates forming in the respiratory tract of patients. And so Pradeep Singh, Neil Toybee, Pete Greenberg, these studies have been shown for a while now. Unfortunately, there are not good animal models for growing these chronic Pseudomonas, or for giving us chronic Pseudomonas infection in like a mouse or a rat, or these animal models that we can use in the laboratory. And so, during my postdoc, I did my postdoc training at Dartmouth Medical School, and there I helped set up a system where we could grow these bacterial biofilms in association with airway epithelial cells. And so my training is actually as an epithelial cell biologist, and at Dartmouth is kind of when I fell in love with microbiology and decided to kind of marry the two in my lab for my own independent program. And so we grow human airway epithelial cells. We can grow them as primary cells, which means we can take patient tissue and culture the cells in a way that they make cilia, and the cilia beat, like I was describing mucociliary clearance. Those cilia can beat in our airway cell cultures, so we can grow them in a way that they look very similar and behave very similar to what they would be doing in the respiratory tract. And then we've developed models where we can then add bacteria to it, either a single bacteria, or in my laboratory, we're really interested in polymicrobial interactions and viral bacterial interactions. So we can combine lots of different microbes in this system to allow us to try to kind of mimic what's going on in cystic fibrosis, and then try to figure out why Pseudomonas becomes dominant, or the bacterial biofilms form, or why they're resistant to antimicrobials. A lot of those questions that we have, we can model in these in vitro systems in our laboratory.
So, okay, what's the, I've been asking all my questions, but what are some of the major questions that you have that you're trying to solve, if I haven't covered them?
Yeah, so So one of our major questions is kind of what we started the lab with, and it's one of those things in science that the more questions you answer, you just have 10 more questions for every one you answer. So we started the laboratory with this observation. I wanted to know why Pseudomonas formed these chronic infections and how biofilms got formed. And in looking at the clinical literature, we saw an abundance of literature that showed that when patients they converted from having acute occasional infections with Pseudomonas to a chronic infection, meaning they had the same infection for decades, that always seemed to correlate with very commonly it correlated with when patients had an acute respiratory viral infection. And so this was shown in many studies, but because of our limitation of not having good model systems, not many people had actually studied what this interaction might mean. And so because I had this system where I could grow these biofilms on respiratory epithelial cells, I thought I could actually maybe ask some questions about, does a respiratory viral infection change the host environment in some way that allows bacteria to form these chronic infections? And so that was the big question we started the lab with, and we're still studying it. So I think there's lots of things we're going to need to figure out, but we've kind of dug pretty deep on mechanisms of how this is happening. And so the answer is yes, respiratory viral infections do promote the biogenesis of Pseudomonas biofilms, and they also promote Staph aureus biofilms. And so we think this could be a general mechanism whereby if you have bacteria in the right place at the right time in your respiratory tract, like if you're a CF patient that does not have effective mucociliary clearance, and you get a viral infection, the conditions change in the respiratory tract in such a way that now the conditions are in place to allow the bacteria to get a foothold and start making biofilms. And once they get to a certain state, now our antimicrobials are no longer effective and they kind of have gotten their foothold. And so we've spent a whole lot of time trying to ask, what is happening during that respiratory viral infection that allows the bacteria to get a foothold? And so one of the big things that we're excited about is this idea of, and I touched on it a little bit at the beginning, of nutritional immunity. And so thinking about the host's ability to sequester nutrients, and it appears that that's getting dysregulated during the viral infection, and there's nutrients available to the bacteria that shouldn't be there, and the bacteria can acquire those nutrients, which is iron in this case, and form biofilms. And so that's kind of led us down now a number of other new questions of exactly how is the iron being trafficked and how is it being dysregulated? Why is it being dysregulated? And then does this provide some way to think about new therapies? So there's iron chelators that are FDA approved. So could we think about using some of those types of therapies as a way of trying to prevent these co-infections? And so that's kind of one mechanistic. Sorry, go ahead.
Yeah, so has it been noticed that CF patients are more prone to viral infections of the respiratory tract, or less prone, or no difference? Yep. And vice versa, if someone gets a viral infection, are they then more predisposed to getting a bacterial one?
Yes. So CF patients don't get any more frequent respiratory viral infections than a healthy individual, but when they get them, they're really bad. So where you and I would have a cold, CF patients often will be hospitalized and they'll have really prolonged viral infections. And then the correlation between respiratory viral infection, most of the correlation has been made with the development of chronic infections. So that is a very well-documented thing in cystic fibrosis. So when patients get respiratory viral infections, they tend to convert to a chronic bacterial infection. And that's often seen with Pseudomonas aeruginosa, but that's also the most common bacteria they have. So that there's some of that could be part of the reason for that correlation.
And when biofilms form, how fast do they form in someone? Does it probably happen, I don't know, within a few days or immediately, or does it take months or years? What's been observed?
But in patients, I don't think we know. So patients will go through. So when a CF patient is born, for Pseudomonas aeruginosa at least, they'll culture negative. When they're first born, they'll culture negative. They'll then pretty early in life, many patients will get a Pseudomonas infection, but the physicians and infection control is they treat it pretty aggressively. So then they'll go through a number of years where they have kind of acute infections and it's able to be cleared with antibiotics, and they kind of so the infections come and go. And then at some point, they will transition to what we consider a chronic infection. And that's when they're going into clinic, they're repeatedly testing positive for Pseudomonas. And if you do, we've now, since we have this next generation sequencing kind of revolution that's happened, if you sequence their isolates over time, it's a clonal lineage of Pseudomonas. So it's the same Pseudomonas that they've had when they start repeatedly testing positive, it's the same Pseudomonas. And so we think that then it is a chronic infection.
Okay, got it.
And then those infections are the ones I said that when we think at that point the bacteria are in biofilms, but we don't know how fast that actually occurs. So when we study it in our epithelial model, because we do live cell imaging, we can watch the process. Within 4 to 6 hours, we have biofilms forming that are that we can't solubilize enough tobramycin or antibiotics to kill. So we know it can happen very quickly, but that's in a cell culture model without, in that particular model, there's no immune cells there. So there is some caveats to that, but I do think it can happen very quickly. It's just the conditions have to be right to allow that to happen. And so we think what the respiratory virus infection is doing is allowing those conditions to be right.
What if there was a mouse model where you had a mouse that had this Pseudomonas that was characteristic of when it's in a chronic infected state, and then somehow either had a healthy mouse aspirate that or put it into their respiratory tract? Has that been tried to see if they immediately get a chronic infection or if it's curable other ways? I don't even know if this happens in mice or can be.
So the problem is we can't get it to happen in mice. And so I think there are so for some things I think mice and small rodent models can be really good models for studying human disease. I think for a lot of respiratory infections, they're not the best model, and particularly for trying to study chronic bacterial infections. And so bacteria and Pseudomonas specifically does not form chronic infections in rodent models. And so some people have gone and they've embedded bacteria in agar beads, which is one method that's used. That doesn't necessarily replicate what's happening in patients, in that patients do not have agar in the respiratory tract, and the bacteria. There's been concern in the field that when you put bacteria in an agar bead, they don't necessarily behave like biofilm bacteria. They just behave as planktonic bacteria that are now growing in an agar bead. And so they don't really replicate the behaviors that they're actually doing in the respiratory tract of a patient with cystic fibrosis. And so we think that probably these mouse models, just until there's some changes in the way, either some genetic changes that we figure out to be able to change the mice in a way that makes them more amenable, they're just not a good model for studying these chronic bacterial infections.
Okay, fine. What about in the lab? Have you tried to culture, let's say, Pseudomonas that was in biofilm form, and then expose it to naive epithelial cells and see if it forms a biofilm faster or if its gene expression is different?
We have not done that exact experiment, but we're about to embark on, I think maybe where you're headed with your question is that some of our questions are, we're really interested in how microbial communities evolve over time and if bacteria get better at making biofilms, which I think is maybe sort of what you're asking. And so we have models now set up that we're starting in the lab to look at if we repeatedly grow biofilms with the same bacteria again and again, do they evolve mutations to get better at making biofilms? And if so, what are those mutations, and can we learn something from those mutations? And so a colleague of ours here at the University of Pittsburgh, his name's Vaughn Cooper, he uses a bead model where he has a polystyrene bead that he has Pseudomonas or Burkholderia senescipacea. He grows biofilms on those beads and he pathogens it daily for weeks and months. And they look at kind of how did the bacteria evolve to make better biofilms over time. And they've done really elegant ecology studies trying to map what are the mutations that evolve over time to make the bacteria better at making biofilms. And so they've gotten really exciting insight on this bead model. We're now working with them to use our cell culture model to try to mimic even better what the host conditions are. And if we can mimic the respiratory mucosal kind of interface even better, what are those first mutations that the bacteria need to be able to try to make a biofilm in the host. And so those are studies that are underway in the lab that we're really excited about. So hopefully people will hear about that soon.
Yeah, you put it in a better way than I did, but Oh, great.
You've been
Well, there's
listening
only,
to the Finding Genius
millions more questions.
podcast with Richard
There's millions of more
Jacobs.
questions to ask and answer. But,
If you like what you hear,
you know,
be sure to
we're close to the end
review
of
and
time.
subscribe to the
What's the best
Finding
way for
Genius
people to find
podcast
out more
on iTunes
about your
or wherever
research?
you listen to
Where can they go?
podcasts.
So
And
my,
want to be
I
smarter
have
than
a
everybody
website
else?
on,
Become
my
a premium
department's,
member at
website. And so
findinggeniuspodcast.com.
if they look at
This podcast is for
if they
information
just
only.
Google
No
Jennifer
advice of any
Bomberger,
kind is being given.
will
Any
bring
action
up
you
my
take or don't
laboratory
take as a
website
result of
and,
listening is
see
your
the
sole
work that we're doing.
responsibility.
Okay,
Consult
great. Well, Jennifer,
professionals
thank you
when
for
advice
coming
is
on the
needed.
podcast. I really appreciate it.
Yeah, I had fun. Thank you for taking the time and talking.